Right? So at this point here, we're now moving on to that last lecture, the review that we hadn't gotten to on exam 4. And if you remember, in terms of the point breakdown, 30% of this exam is geared towards respiratory system. Maybe 5% of that is going to be repeat, questions and information regarding mechanics of ventilation, pressure gradients, pressure differentials for airflow, applying Ohm's law to airflow, and the various types of drugs that can be used to treat asthma as an obstructive disorder, as we just reviewed. And then the rest of that respiratory section on this final exam is going to be new information, testing you on what you learned from the external internal respiration lecture on the transport of oxygen and CO2 and the respiratory centers of the brain, which we did a little bit of review on earlier. So here, in terms of transporting oxygen in the blood, let me blow this up. We know that there are 2 ways to do this: plasma and the hemoglobin. The primary way would be with hemoglobin. Hemoglobin is a protein that functions in gas transport, and it greatly increases blood's oxygen carrying capacity. Okay. By how much? It depends on the hemoglobin concentration. Does the individual have a normal hemoglobin concentration? Great, they have a normal oxygen carrying capacity. Or do they have a higher than normal hemoglobin concentration? That would suggest they might have a higher than normal O2 carrying capacity, lower hemoglobin concentration, or a normal concentration, but an issue with the hemoglobin itself being functional. Then if that's the case, they would have a low O2 carrying capacity. And by definition, that would mean that they're anemic. In those types of situations, we'll talk about in a little more detail towards the end of this review. If we look at how plasma transports gases, one thing to note is which gases have the higher solubility in fluids like plasma fluid. That would be CO2 compared to O2. We're comparing those two gases directly. And what this would mean is oxygen would require a greater pressure to dissolve in plasma. It, according to Henry's law, would need a greater pushing pressure given its lower solubility to produce the same concentration in plasma as the concentration of CO2 if we're comparing the two directly. So the law that we're describing here when asking the question, which gas requires a higher pressure to dissolve in plasma, the law we're using here is Henry's law. We just described that. I also noted, can transport of dissolved oxygen alone meet the O2 requirements of tissues? The answer to that is no, right? Just transporting oxygen dissolved in the plasma is not enough oxygen in order to run our cells. We would need a very high cardiac output. I said impossibly high because no one can maintain a cardiac output that great in terms of turnover of oxygen and the gas exchange that needs to occur at the lungs to get that amount of oxygen to tissues in the body. And so that's where hemoglobin becomes very important. Hemoglobin is a reservoir for oxygen, has high affinity for oxygen, good binding capacity here, and can carry a lot of oxygen directly in the red blood cells by binding to the iron on the hemoglobin. So hemoglobin, in terms of its role that it plays here in increasing our oxygen carrying capacity greatly, you can ask, well, how do we calculate hemoglobin's oxygen carrying capacity? I noted that you're not going to need to actually do this calculation on the exam. You won't have to calculate hemoglobin concentration. You won't have to calculate the O2 content of the blood, but it's useful to know the variables and the units that are used in these calculations. So take note of that, and I have these noted here. So to get oxygen carrying capacity for hemoglobin, we would multiply our hemoglobin concentration, say 15 grams per cent as an example here, by the standard or constant, you could say, of 1.34 milliliters of oxygen. That's the amount of oxygen that can be carried per gram of hemoglobin when hemoglobin is fully saturated with oxygen. So this gives us a sense of the maximum oxygen content of the blood, which I mentioned here. And that's what we think of when we think of hemoglobin's oxygen carrying capacity. I noted, however, we're assuming when we do this calculation that hemoglobin is fully saturated, that one hemoglobin molecule has each one of the heme group ions carrying an oxygen molecule. And we're also assuming in this particular calculation that I've done here to get the 20.1 milliliters of oxygen percent, we're assuming that the person has a normal hemoglobin concentration. Right? And that will give us our normal hemoglobin oxygen carrying capacity. Pay attention to the units here for that oxygen content. Milliliters of oxygen per 100 milliliters of blood, or percent. And of course, that constant 1.34 milliliters of oxygen per gram of hemoglobin makes the assumption that the hemoglobin is fully saturated when we get this volume measurement for oxygen. The 15 grams per cent in the equation above represents a normal or average hemoglobin concentration, grams of hemoglobin per 100 milliliters of blood. So go read through these, make sure you pay attention to the units that are being used here. Zoom out a little bit here. In terms of transporting various types of substances with hemoglobin, or I should say gases with hemoglobin, hemoglobin can carry more than just oxygen. You can also bind CO2, and technically hemoglobin will bind carbon monoxide, and will bind that carbon monoxide better than it binds oxygen. So when oxygen is bound to hemoglobin, right, that oxygen binds to the iron ion in the heme, the Fe2+, ferrous iron. And in that state, we refer to the hemoglobin with bound oxygen as oxyhemoglobin. When it releases that oxygen and is no longer carrying oxygen, we call it deoxyhemoglobin. If instead we have CO present at high concentrations, and that carbon monoxide begins to bind, it's going to bind at the same binding site as oxygen. It binds to the ferrous iron in the heme, and that will produce carboxyhemoglobin. CO2, when it binds to the hemoglobin, it doesn't bind at the iron or ferrous iron binding site within the heme. Instead, CO2, like hydrogen ions, bind to the histidine residues on the globin chains, the alpha and the beta chains. And when CO2 is bound to those histidine residues, we refer to the hemoglobin in that state as carbaminohemoglobin, carrying CO2 in this way. And there were some specific notes I wanted you to keep in mind here about the formation of carbaminohemoglobin. Some of these I go over in the recorded review at the very end in terms of the ways that CO2 is transported in the blood in the lecture on internal respiration. And here I ask, well, how does the carbaminohemoglobin form? What is the site of attachment? The globin chains in those histidines on the globin chains are where that CO2 attaches to the hemoglobin. I know that this is one of the ways that CO2 is transported in the blood, but is it the main way it's transported in the blood? The answer is no, it's not the number one way it's transported in the blood. The primary way that CO2 is transported in the blood is in the hydrated state. And this just refers to the buffering reaction that occurs when CO2 combines with water to produce carbonic acid that dissociates and produces bicarbonate and hydrogen ions. So most of the CO2 that dissolves in plasma will undergo this process of the buffering reaction. And most of the CO2 that dissolves in the plasma but then diffuses into the red blood cells will also undergo that process. That's the main way that CO2 is being carried. But some CO2 does bind directly to the histidines on the hemoglobin in the red blood cells. And that's what we're describing here when we say carbaminohemoglobin. Now, sticking with this general information about transporting CO2, I ask, what are the steps when CO2 is picked up in the tissues? And so you want to run through those steps here. The CO2, when it's being picked up by the tissues, it's diffusing into the plasma. And in the plasma, various things can happen to it. The primary thing that happens is that it undergoes this hydration. But a portion of that CO2 will diffuse into the red blood cells, and various things can happen there to the CO2 to help it be moved and transported, but it's primarily going to be hydrated there. When we do get that hydration occurring and we produce carbonic acid that dissociates to hydrogen ions and bicarbonate, the hydrogen ions can be picked up by histidines on the alpha and beta subunits of hemoglobin to help buffer the hydrogen ions within the red blood cells. The bicarbonate moves out into the plasma. So that's what I noted here. Where does the bicarbonate end up during this process? It ends up in the plasma. The more CO2 we have dissolved in the plasma, greater our PCO2. The more hydration of that CO2 will occur with our buffering reaction, the greater bicarbonate concentration we produce in the plasma. And what I'm describing here with that relationship is just a very basic rule for Le Chatelier's principle of how the buffering system works. Buffering system is shifting to oppose a change. We've loaded up on CO2, right, at the tissues, and we get this buffering reaction where we're now bringing down the levels of CO2, but producing more bicarbonate to help balance that increase in CO2 that's occurred here. And then what are the steps when CO2 is dropped off at the lung capillaries? Well, CO2 is diffusing into the lung space, and all of these reactions will go in reverse, where the bicarbonate then combines with hydrogen ions, produces carbonic acid that dissociates to produce water and CO2, which then diffuses out of the red blood cells, out of the plasma, into the airspace. And during that process, we'll see, according to Le Chatelier's principle, this reaction will go in reverse. The PCO2 begins to decline. And in order to bring up that level, we get more bicarbonate moving into red blood cells, combining with hydrogen ions and causing that buffer reaction to move in the opposite directions. It's what's driving the formation of CO2. And that is what's driving the diffusion of CO2 out into the lung space, is this constant production of the CO2 with that buffering reaction moving in the opposite direction. All right, within the lecture itself, before we get to the various ways that CO2 is transported within the blood, we look at the hemoglobin's affinity for oxygen. And here I wanted you to review hemoglobin's affinity for oxygen with respect to cooperative binding and loading at the lungs and unloading at the tissues. So I just wrote the basic notes here that are important for understanding the loading of oxygen onto hemoglobin, the unloading of oxygen at the tissues. So here I say during the loading process, this is happening at the lung capillaries. And at this time, hemoglobin has a very high affinity for oxygen. When hemoglobin's fully loaded with oxygen, all 4 iron ions, ferrous iron ions, are carrying oxygen. And that's when hemoglobin has the highest affinity for oxygen overall. Right? That's part of the cooperative binding process. the fact that the oxygen is being loaded onto the hemoglobin is occurring because of the partial pressure gradient that exists here between the partial pressure of oxygen in the alveolar space and the partial pressure of oxygen in the capillary fluid. So we look in the alveolar space and we see a partial pressure of 100 millimeters of mercury. In the capillary fluid, the plasma fluid coming to the lungs from the body, we're at 40 millimeters of mercury for partial pressure. That differential favors oxygen diffusion into the plasma and the loading of the oxygen onto the hemoglobin. Once we fully saturate the hemoglobin during transit time, then we have the remaining oxygen diffusing into the plasma, dissolving in the plasma until we reach a partial pressure in the plasma of 100 millimeters of mercury. And then we get no further net diffusion. But at this point, we should see that the hemoglobin is fully saturated. All of the iron ions are carrying oxygen. When that loading occurs, the Fe2+, the iron ion, it goes into a relaxed state. Physically, it goes into a relaxed state within the heme. It falls below the heme ring, the iron ion, when it's holding the oxygen. In that way, we call it a stable configuration. And when that occurs, that actually causes physical change to the alpha and the beta polypeptide subunits surrounding that iron ion and heme, and that pushes the CO2 and the hydrogen ions off here within the lung capillaries. So we say we remove the CO2 and the hydrogen ions from the globin chains, the hemoglobin chains, and the removal of those acids, if you will, is called the Haldane effect. The hemoglobin is acting like an acid, especially because it's releasing the hydrogen ions. It's dissociating hydrogen ions into solution when that CO2 and the hydrogen ions are being removed. So that's sort of the progression of the physical interactions that are happening here during loading of oxygen with the lung capillaries and the pressure differentials that we would see associated with that loading of oxygen. Then at the tissues, the pressure differential that we see there is that in the arterial blood, the partial pressure of oxygen is around 95, 97 millimeters of mercury because of the oxygen that's loaded on the hemoglobin at this time. And the more so importantly here for the partial pressure, the amount of oxygen that's dissolved in the plasma is still high. But the amount of oxygen that's dissolved in the interstitial fluid and that associated with the cells is relatively low at 40 millimeters of mercury because the oxygen is constantly being depleted by the body cells. So this differential favors diffusion of oxygen from the blood to the tissues. And that oxygen is being released during this time in this cooperative unbinding or unloading process, because hemoglobin now has lower affinity for that oxygen. And that lower affinity is occurring here, that decreased affinity is occurring because here at the tissues, we have CO2 and hydrogen ions beginning to attach to the globulin chains, attached to those free histidines that are available. The hemoglobin is effectively taking hydrogen ions out of solution. And that's referred to as the Bohr effect as that's happening because the hemoglobin's acting like a base by removing the hydrogen ions and buffering those hydrogen ions. And because of the attachment of CO2 and hydrogen ions, that causes a physical change in the alpha and the beta subunits. That causes then a physical change in the position of the heme group and the iron ion that's holding the oxygen. We say that the iron goes into a tensed state. It moves above the ring as these physical changes are happening, and the whole structure becomes wobbly. And that makes it more likely that the iron ion will let the oxygen go. We say iron ion has decreased affinity. It's all because of these physical interactions that are occurring here with the metabolites like CO2 and hydrogen ions. In terms of the affinity for hemoglobin, I want you to know how hemoglobin's affinity for oxygen changes with the number of oxygen molecules that are bound to a hemoglobin. And we see evidence for the fact that hemoglobin's affinity for oxygen actually changes because of the sigmoid curve that's associated with the release of oxygen to the tissues. What we say here is there's allosteric binding that shows us that hemoglobin has a changing affinity for oxygen as it binds and unbinds those oxygen molecules. So most useful thing to note here is that the hemoglobin has the highest affinity for oxygen when all 4 subunits of hemoglobin are bound to oxygen. This means that it's hardest to remove the first oxygen. We need a very big pushing pressure, if you will, to get that first oxygen off. But once that first oxygen has been removed, it then becomes easier to remove the second and the third oxygen. Because after the first one is off, now the hemoglobin molecule overall has a lower affinity for the remaining oxygen molecules. So the second and third now will be easier to remove, less of a pushing pressure to get the second and third off. Once we only have one left, the hemoglobin molecule has a slightly increased affinity for that last oxygen molecule. It's going to be slightly harder again to remove that last oxygen. We need a little more pushing pressure than we did with the second and third. The pushing pressure is referring to the partial pressure gradient for oxygen that is required. We compare the partial pressure in the lungs to that of the tissues. We ask, how big does that pressure differential have to be in order to remove that first oxygen and the second and third? That gives us an indication of just how great hemoglobin's affinity is for the oxygen, is whether there is a big pressure differential or smaller pressure differential. And then in terms of being able to read the oxygen hemoglobin dissociation curves here, I said review the process of reading the diagrams, both the oxygen saturation axis here, the percent oxyhemoglobin saturation axis, that tells us how many oxygen molecules are being carried by the hemoglobin. If we're at 100% saturation, all 4 of those subunits are carrying oxygen. 50%, 2 of the 4 are carrying oxygen. So we're going here. And also look at the oxygen content axis. So use those diagrams that have both axes. There are examples in the homework packet that would be useful to review. I pointed out just generally the questions that you should take a look at in the homework packet 4. Questions 29 through 43 are good ones to look at. Some of them involve oxyhemoglobin dissociation curves, so especially take a look at those and make sure you can read them appropriately. And also look at the CO case study that was in the last pages of the lab manual. That one in particular, we're using the carbon monoxide issue the individual is experiencing, comparing that to normal oxygen carrying capacity and normal oxygen content and the normal amount of oxygen that can be delivered to tissues. These are some questions actually that came from the homework packet that I wanted you to take note of. For instance, will you get additional loading of hemoglobin if you increase the partial pressure of oxygen here, say on this diagram, to 300 millimeters of mercury versus the 100 millimeters of mercury? 100 millimeters of mercury is the PO2 we expect during loading at the lungs. If we go out to 300 millimeters of mercury, and we look at whether or not we can saturate that hemoglobin any further with that increase in pushing pressure, we're already at 100%, right, saturation of hemoglobin. So whether we were at 200 or 300 millimeters of mercury, that's not going to make a difference here. We're already at 100% saturation. We can't saturate the hemoglobin anymore. But we can do something by increasing the partial pressure to 300. We can push more oxygen into the plasma. dissolve more oxygen into the plasma. And technically, we could deliver more oxygen to tissues by doing this. But it would be the plasma component that has a greater concentration of oxygen rather than more oxygen being carried by the hemoglobin. So these were a few questions, multiple choice questions that were specified on the homework packet for. Take a look at there. Another scenario question I have here. What if a patient has half the normal hemoglobin concentration rather than the 15 grams percent? Let's say they have 7.5 grams percent. We can ask what would happen to their percent oxyhemoglobin saturation? Would it scale down? What's going to happen to their oxygen content? Well, we know what would happen to their oxygen content because we can do just a quick calculation here to determine what the oxygen carrying capacity of their blood would be, the hemoglobin. If they have 7.5 grams percent for hemoglobin concentration, and we assume they're fully saturating their hemoglobin during that exchange at the lungs, we just multiply 7.5 by the 1.34 milliliters of oxygen per gram of hemoglobin to get the maximum oxygen carrying capacity for this individual's hemoglobin. And it's probably going to be around the 10 that's shown here on this diagram compared to the normal 20. And so their overall oxygen content is going to be greatly decreased. But my question here was, what would happen to the percent oxyhemoglobin saturation? What would happen here to this scale? And the answer here is, unless I tell you otherwise, we're assuming that they can fully saturate their hemoglobin when they have gas exchange at the lungs. And so even though they have 7.5 grams percent for hemoglobin, when exchange happens at the lungs, they're still fully saturating that smaller concentration of hemoglobin. They'll still have 100% saturation. So this axis here would have to scale down for the individual that has the lower oxygen content. And we'd say maximum they can carry 10 milliliters of oxygen per 100 milliliters of blood, or mlO2%. And at that maximum, they're still fully saturating their hemoglobin. So we go across here and we'd see they'd be at 100%. So we'd scale this axis down for that individual. And I'm going to show you a diagram like that on, an upcoming slide where we redraw this diagram to show what that would look like for an individual that has, in this case, anemia. They have a low oxygen carrying capacity, right, with 1/2 the normal hemoglobin concentration. With this in mind, unless it's stated otherwise, right, we assume they can fully saturate their hemoglobin. We can think of, well, what would be a scenario where we wouldn't make that assumption that they can't fully saturate their hemoglobin, right? That would be the scenario where I tell you the individual has an obstructive or restrictive disorder. They can't exchange gases appropriately at the lung epithelium. And if that can't occur across the airspace and the alveoli and the plasma, then during transit time, we might not see full saturation of the hemoglobin. We might not be at 100%, right? How can we indicate that? Well, that's where a pulse oximeter becomes useful, because that tells us how fully saturated the hemoglobin is, and nothing more than that. Another way we could have an issue with an individual being able to fully saturate their hemoglobin is if they have a cardiovascular disorder, okay, heart weakness or what have you, where they can't effectively perfuse their lung capillaries. And so during that transit time, they don't have the normal volume of blood coming to the lungs. So thus they don't have the normal amount of plasma and hemoglobin available for gas exchange to occur. And so they're going to have a lower level of hemoglobin saturation because of that. All right, so we're almost done with this section here. Still thinking about hemoglobin's affinity for oxygen. I wanted you to think about the factors that affect that affinity for oxygen and whether they would cause a left or a right shift on the graph. Always keep in mind that a right shift means that overall hemoglobin has a decreased affinity for oxygen. A left shift means that overall hemoglobin has an increased affinity for oxygen. The first factor I have here is 2,3-DPG. 2,3-DPG is a byproduct of red blood cell lactic acid fermentation. I mentioned that here. And we get an upregulation of 2,3-DPG production with an increase in metabolic rate, or when an individual is experiencing hypoxia, we might see that that amount of lactic acid fermentation in red blood cells increase as well. The 2,3-DPG as a substance can bind to the globin chain histidines in the same way that hydrogen ions and CO2 can. And it will produce the same changes, physical changes, that help push oxygen off. They put the heme and the iron ion into the wobbly state, the tense state, that decreases the affinity that hemoglobin has for oxygen, makes it easier for it to be removed to the tissues. So when we increase levels of 2,3-DPG, we expect a right shift compared to normal. Decreased levels, a left shift. No, overall, when we think of changes to 2,3-DPG, increases in those levels are typically referring to more metabolically active tissues or instances where an individual might be experiencing hypoxia. Temperature shifts, those shifts, let's see, if they increase in temperature, that's indicative of increased metabolic rate. Decrease in temperature, decreased metabolic rate. And so when we have an increase in temperature, we expect with a greater increase in metabolic rate, we'd have more CO2 production, more hydrogen ion production, with 2,3-DPG production, and that would give us a right shift. We have more of those metabolites to help push oxygen off. Hemoglobin is going to have a lower affinity for oxygen overall. Decrease in temperature, decreased metabolic rate, less of those metabolites being produced, and we'll see a left shift on our oxyhemoglobin saturation curve. And then for hydrogen ions and CO2, when we have an increase in hydrogen ion concentration, which equates to a decrease in pH, right, more acidic pH, we expect a right shift. Same is true when we have an increase in the partial pressure of CO2, more CO2 being produced as a byproduct of aerobic respiration. Is that right shift? And then the opposite for low hydrogen ion concentration, which represents a basic pH, and low partial pressure of CO2. So generally, yes, we're getting more of these metabolites, right, when metabolic rates have been increased, or keep in mind, hypoxic conditions, as I mentioned earlier, would lead to a buildup of these substances. And in this way, we can think of it with respect to perfusion in Unit 3, where we discussed active hyperemia and reactive hyperemia, the types of local signals that will cause vasodilation to occur. When we have more metabolically active tissues, we have more CO2 being produced, greater hydrogen ion concentration, right? Drop in pH is occurring there. And that's a signal that causes arterioles to relax, vasodilate, and the precapillary sphincters to relax. And we have greater blood flow to the tissues. And so that process is happening, thus bringing more oxygenated blood to these tissues that have the buildup of metabolites, right? That's happening in one respect with active hyperemia, when the tissues are just more metabolically active. But we could see the same type of effects if an individual has had blockage of blood flow, some sort of hypoxic conditions, ischemic conditions there for the tissues, And then when blood flow returns, we get that reactive hyperemia where we've had buildup of these metabolite substances, not because the tissues were just more metabolically active overall, but just because there was a lack of blood flow for a period of time when there should have been blood flow. So those slight differences are useful to keep in mind, that not only might we have just normally functioning tissues that are metabolically active and have a high metabolic rate, but something could have gone wrong in terms of blocking blood flow, leading to the same responses. But part of the same response here would be we get more oxygen released to the tissues because we have more of these metabolites present under those conditions where there's been a blockage of blood flow. Now, problematic one here is the CO, the carbon monoxide. It, let's fix that here. CO increases when we have an increase in the partial pressure of CO. And we should typically have a low partial pressure of CO within our tissues because there's very little within the actual atmosphere that we're breathing in. But should we be in an environment with high levels of CO in the atmosphere, then that concentration gradient or that pressure gradient will drive the flow of CO into our blood plasma and cause CO to load right on our hemoglobin in the same way that oxygen does and displace that oxygen. And what this produces then on our oxyhemoglobin dissociation curve is a left shift. And there's two reasons for this occurring here. Hemoglobin's oxygen binding or carrying capacity overall is decreased. Less oxygen is being bound if more CO is being bound and is being carried in the same way that the oxygen would be. At the same time, hemoglobin actually has an increased affinity for the oxygen it has already bound. So I know that here, hemoglobin's affinity for bound oxygen actually increases. And so it's holding on to the oxygen it's already carrying more tightly when CO is present. And that produces the left shift on our curve. Then we have the example of fetal versus adult hemoglobin that I want you to pay attention to. And really the shift that we see here depends on your perspective and the way the question is worded. So pay attention to that on the exam. So there will definitely be a question where you have to do a comparison here between fetal and adult hemoglobin and the shift that arises. So if we focus on the fetal hemoglobin versus the adult, so compared to the adult hemoglobin, fetal hemoglobin shows what type of shift. It's a left shift. And that's because there's decreased binding of fetal hemoglobin of 2,3-DPG. And that's due to the gamma chains that are used rather than the beta chains. Gamma chains have fewer, have a lower affinity for binding 2,3-DPG. If I ask you compared to fetal hemoglobin, the adult hemoglobin shows what type of shift compared to the fetal. It looks like the adult is shifted to the right. So we say a right shift. The beta chains do bind 2,3-DPG. And this way that will give us overall a decreased affinity of hemoglobin for oxygen. So the extra note I made here is we often say that fetal hemoglobin has a higher affinity for oxygen compared to adult hemoglobin, but how we describe which curve shifts depends on the context of the question. What is our starting point? Who are we comparing that starting point to? The example I give here is that if I say all else being equal, right, the amount of hydrogen ions we have present, the partial pressure of CO2, that's equal for the fetal and the adult hemoglobin in this scenario. And if we add 2,3-DPG, which curve actually shifts? The one that's going to shift is going to be the adult curve. It's going to shift to the right compared to the fetal curve, because we've now added this extra substance that helps to decrease hemoglobin's affinity for oxygen even further. All right. A few more scenarios here. This one is one that actually has a connection between the respiratory system and the buffering information we went over as part of the lab lecture for acid-base balance, thinking about Le Chatelier's principle once again. But here, I want you to think about how the PCO2 changes with hypoventilation, hyperventilation. What would be the effects on the bicarbonate buffering system when we had hypoventilation and hyperventilation, right? And then extend that to the effects we would see for the oxyhemoglobin saturation curves, right? And just keeping in mind that when we say that hemoglobin acts as an acid in lung capillaries, why do we say that? Well, it's because the hemoglobin is releasing CO2 and dissociating those hydrogen ions. That way it's acting like an acid. And in the tissues, hemoglobin, we say acts as a base because it's taking hydrogen ions, CO2, out of solution. So here I'm writing the buffering equation, keeping in mind that for a simple buffering system, we're just following what we call Le Chatelier's principle, where we see a shift in the buffering system, the direction of that chemical reaction to oppose the initial change. So let's start with the scenario of hypoventilation. When an individual is hypoventilating, decreased rate and depth of breathing, they're trapping more CO2. What is happening to their plasma concentrations of CO2? Those concentrations are plasma partial pressure of CO2. Those levels are increasing. So we see an increase in PCO2 when the individual is trapping CO2. As PCO2 increases, right, our bicarbonate carbonic acid buffering system is going to respond in such a way to bring down those levels once again. Help balance that increase in PCO2 by shifting to the production of more hydrogen ions in bicarbonate. So we see hydrogen ion levels increase, bicarbonate concentration increase. That's going to bring down PCO2, raise bicarbonate, trying to balance those levels of acid and base once again. So while the PCO2 has been rising with this buffer shift, we see the bicarbonate levels in the plasma begin to rise. Right? So that would be the effect on the bicarbonate buffering system, that we see bicarbonate levels rise. We're also seeing the hydrogen ion concentration rise, right? And in terms of what's happening at a tissue level for our release of oxygen to tissues, while the individual is trapping CO2 and their PCO2 is rising, and at the same time, right, as part of the buffering reaction, the hydrogen ion concentration is rising. Both of those are metabolite signals that causes a shift in our oxyhemoglobin saturation curve. High PCO2, higher hydrogen ion concentration, a shift to the right. Becomes easier to unload oxygen to the tissues in this scenario. And we see just the opposite here with hyperventilation. An individual here is hyperventilating. They've increased their rate and depth of breathing above and beyond the what's required based on metabolism. And so they're losing more CO2 than they're producing via metabolism. So their partial pressure of CO2 within their body fluids begins to decline. According to Le Chatelier's principle, to oppose this change, this deficit of CO2, the buffer shifts to the direction of trying to increase levels of CO2. So we have bicarbonate and hydrogen ions combining to produce carbonic acid that dissociates, raising levels of CO2. And during this process, we see the individuals experiencing low PCO2, and with the removal of bicarbonate and hydrogen ions, as this buffer shifts, we see low levels of hydrogen ions. Low levels of hydrogen ions, low partial pressure of CO2, metabolite levels that we relate to a left shift on our oxyhemoglobin dissociation curve. And for the tissues, we see that it's harder for the hemoglobin to unload oxygen to those tissues while the individual is experiencing this hyperventilation. And that's merely because now we have less CO2 and fewer hydrogen ions in order to cause that wobbly state, that tense state, to help push oxygen off compared to normal. So I like this one. I mean, it's a stretch, but it's true. This is a scenario that we would see where we're now relating these respiratory issues and the buffering system that is part of this this relationship here, with the actual metabolites, PCO2, hydrogen ions are part of the action that leads to loading, unloading of oxygen. The last 2 points here that I have refer to diagrams. And I've drawn some examples of these diagrams here. One was, I want you to think about what the oxyhemoglobin curve would look like for somebody who has anemia. So I mentioned that earlier, that we come back to an actual diagram of this. I want you to think about both axes. With anemia, that individual by definition has a reduced oxygen carrying capacity. That means that they have reduced O2 content. So rather than the 20.1 milliliters of oxygen percent, let's say that their hemoglobin concentration was half that. So they have half the oxygen content, around the 10 milliliters O2 percent. So that's what I drew here. That's their starting point in terms of their oxygen content. But if we're assuming that they have no other disorders, respiratory, cardiovascular, then they should be able to fully saturate that smaller concentration of hemoglobin, right, within their blood. So we see a reduction here in the height of the percent oxyhemoglobin saturation axis. The 100% saturation we have to bring down to scale correctly with their oxygen content. They're fully saturating their 7.5 grams of hemoglobin to get that oxygen carrying capacity of about 10 milliliters O2 percent. And then we can draw our oxyhemoglobin saturation curve. We compare that to this scenario here, where we're talking about an individual who has CO poisoning. So we're noting both axes once again. So this individual, they have a reduced oxygen carrying capacity, reduced oxygen content, because they're displacing their ability to carry oxygen with the CO. They're carrying less oxygen if they're carrying more CO, right? But that also means they have a reduced oxyhemoglobin saturation. If they have 50% saturation of their hemoglobin with CO, then they're only 50% saturating that hemoglobin with oxygen. So we actually don't change the oxyhemoglobin saturation axis. That stays the same, but the individual's oxygen content is lower. So let's say they're at 50%. Even though their hemoglobin concentration might be normal, because the CO is displacing oxygen here, the O2 content is half of what it should be. So that's how we would start our oxyhemoglobin dissociation curve. We'd start it from that point at 50%, about 50%. But we change it somewhat. You look at the curve here and the curve here, and I'm showing that there's a left shift as well. we show that overall hemoglobin has a much higher affinity for that oxygen at every step along the way in terms of the unloading process. So that left shift, right, is because hemoglobin has increased affinity for bound oxygen. Now, I mentioned this a couple times at this point in some of the points earlier, or questions earlier, prompts. That's something you don't want to confuse on the exam, that the oxygen carrying capacity with CO poisoning is reduced. Hemoglobin's oxygen carrying capacity is lower, but hemoglobin's affinity for bound oxygen that's already bound to hemoglobin, that is higher, that is increased. You go opposite directions there. Don't mix them up on the exam. The last thing I note here is you want to review that CO poisoning in the multiple choice questions from that lab manual clinical correlation. I already have the key posted for that, so I don't think that's something we need to review in detail together. And I know I posted Dr. Street's recorded overview from YouTube as well, so that might be useful if you want to run through it with her video too. And of course, come to me with any questions you may have about the oxy-hemoglobin dissociation curve that's shown there, or any of the other questions about treatments or what have you. But expect at least a few questions actually directly from that case study on this exam. So don't forget to review that, okay?